you so much for that introduction, Aaron. Yeah, my name is Brian Kwee. I am a new assistant professor in the Department of Biomedical Engineering at University of Delaware, same department as Stephanie Cohn, who presented last week. And what I wanted to do today is to present to you some of the previous work I did in graduate school and my postdoc, as well as describe how that's motivating the future work I'd like to do at my laboratory. And so a lot of the work I did in the past and what I'll be doing in my lab in the future focuses on tissue engineering. And tissue engineering consists broadly of taking engineered biomaterials that can mimic extracellular matrix found in our body and combining that with ex vivo cultured cells and drugs such as cytokines, growth factors, and small molecules, and combining them to create these cell and or drug containing biomaterials. And you can broadly use these biomaterials for kind of two purposes. One is that you can utilize and grow these biomaterials into human tissues to model human healthy and diseased tissues in vitro with greater fidelity than if you were to study diseases in mice or on 2D tissue culture plastic in vitro. Or you can take these biomaterials with cells and drugs and transplant them in vivo to treat a variety of diseases and injuries where these biomaterials can induce host tissues to regenerate, or the cells that you transplant themselves can be used to replace the function of diseased or lost tissue in vivo. And so a lot of my previous work kind of worked on subsets of this diagram over here. And previously, I did my graduate school at Harvard University and my postdoc at the US FDA. And at Harvard University, I worked in David Mooney's lab, where I mainly worked on drug delivery biomaterials that can modulate the immune system. And specifically, what we wanted to do was to design these biomaterials and plant them in vivo to manipulate the immune system to promote vascular and muscle regeneration, due to the fact that inflammation and the immune system is broadly important for wound healing. Later on in my postdoc, I worked on two other main projects. One project involved combining cells with biomaterials to create these cell and biomaterial combination therapies. But one thing that we appreciated at the US FDA is that the cells that we're combining with these biomaterials are very heterogeneous in terms of cells derived from different donors or heterogeneity on the single cell level. So what we wanted to do was to sort and identify within this heterogeneous population cells of high potency that could be combined with biomaterials to overall improve the function of these cell and biomaterial therapies. And lastly, another project I worked on at the FDA involved combining cells with biomaterials to create in vitro models of lymphatic tissues, specifically on the lymph node. So what I'll do for these next few slides is briefly go over these three different projects that I worked on in the past. So this first project, as mentioned to you before, I worked on immunomodulatory drug delivery biomaterials for vascular regeneration. And the biomaterial that we want to utilize was a synthetic PLGA scaffold that's macroporous. And what we wanted this to design this biomaterial to do was to provide control release of a drug called an antigen, which is an inflammatory cue for recruiting antigen-specific T cells. And specifically what we wanted to do was to recruit antigen-specific T cells that were generated from vaccines that can specifically create T cells that are pro-regenerative or can induce wound healing. And as you can see on the top here, we were able to design this biomaterial to provide sustained release of our antigen ova over the course of 14 days. And our overall working hypothesis was that delivery of an antigen from a biomaterial, specifically our PLGA scaffold, could recruit antigen-specific Th2 T cells generated from alum vaccines to ischemia injury sites to promote vascular regeneration. So in other words, can we use this drug delivery biomaterial to specifically recruit immune cells that are conducive for regeneration, specifically Th2 T cells, the sites of ischemic injury to promote vascular regeneration? And so the following slide here is briefly describing our experimental setup, where we took wild-type mice. We vaccinated them twice with that ova alum vaccine that I mentioned to you before. And then we experimentally induced ischemia in one of the limbs of the mice by ligating the femoral artery and vein, and then implanted a scaffold containing ova on top of that ligation site, where the idea was that this scaffold could recruit Th2 T cells to this ligation site to induce vascular regeneration. And so we had 3 different conditions with varying combinations of vaccine plus PLG scaffolds. We had one group in red here, a naive mouse with no vaccine plus an ova-releasing scaffold. We also had mice that received the full vaccine but contained a scaffold that was blank, had no ova. And in blue here, we had the full combination therapy with the ova plus alum and our ova-releasing scaffold. And what we showed is that in our ova plus alum containing scaffold with the vaccine, that we were able to enhance the recruitment of Th2 T cells to the site of ligation. And furthermore, when we analyzed the ability of these scaffolds to enhance perfusion or induce vascular regeneration across this ligation site, when we looked at the right here, the ischemic perfusion ratio returning over the course of 14 days, we found that our combination therapy with the vaccine and ova overall led to the greatest amount of perfusion coverage relative to our 2 control groups. Overall showing that our drug delivery biomaterial could indeed recruit immune cells that were specific for vascular regeneration and enhance the recovery and blood vessel regeneration at this ischemic site. The second project that I'd like to talk about briefly is the FDA project I was talking about, where we want to identify cells of high potency within a heterogeneous population that could be combined with biomaterials. Where the biomaterials that we wanted to test was a collagen and fibrin hydrogel. These are naturally derived biomaterials that are kind of widely used in clinical trials. And the cell types we want to combine with these biomaterials are mesenchymal stromal cells. And we specifically want to evaluate their ability to suppress T cell proliferation, specifically because MSCs are widely used in clinical trials to treat a variety of inflammatory diseases and autoimmune diseases. And we hypothesized that MSCs of high immunosuppressive potency, specifically on these different biomaterials, collagen and fibrin, could be identified by integrin expression, where integrins are the receptors on cells that allow them to bind to underlying biomaterials. And so to do this, what we utilized was a method called fluorescence-activated cell sorting, where we took unsorted cells here in gray, and we plotted one integrin here on the y-axis, one integrin on the x-axis here, where each one of these dots represents a different cell. And we found that our unsorted MSCs were pretty much 100% positive relative to our negative control for both of these integrins. And then we used stack sorting to sort them into low, medium, and high integrin-expressing cells. And we took these MSCs and cultured them on the biomaterials, and in a co-culture system, evaluated their ability to suppress T cell proliferation. And in this graph, basically, the lower the bar, basically the greater immune suppressive capacity of these MSCs. And what we found was that with increasing integrin expression, there was an enhanced ability of these sorted MSCs to suppress T cell proliferation, where our high integrin expressing cells had better immune suppressive capacity than our unsorted cells. And this overall demonstrates that you can utilize the magnitude of integrin expression specific to a biomaterial to identify MSCs of high immune suppressive function on different biomaterials. The last project I wanted to talk to you about was, again, engineering a lymph node in vitro model. And for our biomaterial, we used a collagen fibrin composite hydrogel, so an interpenetrating network of these 2 naturally derived biomaterials that I mentioned to you before. And the cells that we want to incorporate into this biomaterial were fibroblastic reticular cells. These are stromal cells that are found in our lymph node and that have a function of basically recruiting and secreting cytokines that can regulate the trafficking of T cells and dendritic cells, as well as secreting factors that could regulate the survival and activation of these immune cells as well. So our overall hypothesis was that culturing these FRCs, fibroblastic reticular cells, in this collagen fibrin composite hydrogel could create a model of the lymph node paracortex, specifically the T cell zone, which could overall regulate immune cell trafficking and migration. And to do this, what we did was we actually designed a microfluidic device, as you can see here on the left, in which you can see here this microfluidic device has different channels that are separated by these micropillars, in which you can inject into each one of these channels a different hydrogel containing different cells or constituents. And in 3 of these channels, we basically had one channel with our biomaterial containing these FRCs, which we allowed to self-assemble into a network. We then had a blank hydrogel in the middle here, and then another hydrogel that contained T cells and dendritic cells. And we wanted to evaluate was how these FRCs secrete cytokines that can basically recruit these T cells to migrate across this middle channel and into the FRC channel, kind of as they do in vivo, where these T cells migrate into an FRC paracortex network. And the following diagram here on the right is basically showing confocal images of our 3D network, where we have our FRCs kind of colored in white here for F-actin, where we can also see our FRCs secreting fibronectin, an extracellular matrix molecule, into the surrounding hydrogel. And we see that indeed that our FRCs were recruited T cells from this lymphocyte channel into the FRC channel as a function of chemokines that they're secreting. And you can overall see that these T cells are not only migrating to the FRC channel, but binding and adhering to the FRC network. This overall demonstrates that we can indeed, in this microfluidic device, engineer a kind of lymph node on a chip that could kind of recapitulate the ability that these FRCs have in recruiting immune cells. And so basically, in my future laboratory, I like to continue studying these themes of tissue engineering, immunoengineering, and cell manufacturing, and kind of basically extend these 3 areas of research that I just described to you before. We like to continue designing cell and drug delivery biomaterials that can be implanted in vivo, that can promote pro-regenerative inflammatory microenvironments, where these drug delivery and cell delivery biomaterials can basically control the number and types of immune cells recruited at a site of injury to promote regeneration. We also like to continue applying this sorting approach that I described in my second project, where we can take a heterogeneous population of cells and sort them by the magnitude of integrins as well as cadherins, where cadherins are receptors that mediate cell-cell interactions, and combine these cells with biomaterials for tissue engineering applications that can be implanted in vivo to promote regeneration. And then lastly, we'd like to continue designing in vitro lymphatic tissues, including both lymph nodes as well as lymphatic vessels, where we combine stromal cells and immune cells with synthetic biomaterials that we can rationally design to mimic both the physical and chemical properties of these lymphatic extracellular matrix tissues. So yeah, that's it for the presentation I showed to you for today. I hope that you got a good sense that my lab is working on kind of broadly areas of tissue engineering, immunoengineering, and cell manufacturing, specifically working in the realms of engineering lymphatics, creating immunomodulatory biomaterials, and cell sorting for tissue engineering. And FYI, my lab is located on the STAR campus at the AP Bio Innovation Center on the 4th floor. And this is my email in case you are interested in reaching out, and I'd be happy to take any questions. Thank you.